Welcome to the second lesson of the course. In the previous lesson, you learned about exome and whole genome sequencing. In this lesson, we'll see how we can analyze these datasets. So, we can look at a very basic uh, sequence analysis workflow. First step of a sequence analysis is called the mapping step. The mapping step takes two input datasets, raw sequence data or reads coming from the sequencing machine and a reference sequence. So what is exactly a reference sequence? Reference sequence is a standardized high quality sequence that represents an individual genome. We can think of it as a baseline sequence that is used to compare our input data and create a map of the input dataset. So during the mapping process, each read is mapped or aligned to the reference sequence in a way that the algorithm finds the best possible match for each read. So this process creates a file called alignment file. The next step of a workflow is to refine this alignment file. For example, the alignment file can contain duplicated reads that are duplicated during the sequencing process. These reads can increase biases in the downstream steps, so they need to be marked as duplicated reads. Moreover, the alignment file can contain systematic biases introduced during the sequencing process. So, there are tools that help us to identify these systematic biases and recalibrate the quality scores of the sequences and apply them onto the alignment file. So, the refinement stage perform these steps and create a refined alignment file. This goes to the next stage of a workflow, which is the stage that identifies DNA or changes in the alignment file. This variant detection is one of the crucial steps in a variant calling workflow. We know an alignment file contains the reference sequence and the reads that are mapped to the reference sequence. When we go through these alignment files, we can see that certain bases do not match to the reference base. So these differences are the variants. However, in real life or when running a variant calling pipeline, identifying variants is not this simple. There are advanced statistical methods or machine learning based methods to remove the noises in these alignment files and identify true variants that can be used in clinical or research interpretation. Now we have a basic understanding of what's happening during a variant calling analysis workflow. To implement a workflow, we need to run a set of tools. If we think about a best practices NGS analysis workflow, we can think of several tools to run the workflow. We can use a tool like BWA for mapping and sorting the alignment file that are coming from the initial map, and then to do the refinement we can use the genome analysis toolkit GATK tools like mark duplicate, base recalibrator or apply BQSR. These are the tools that help us do the refinement and create a refined alignment file. Once we have a refined alignment file, we can use a variant caller like haplotype caller to identify SNVs and indels. Now you are familiar with a very basic variant calling and variant calling workflow and the tools that are used to run a variant calling workflow.